Good morning, everyone. Welcome to the Arthritis web seminar series. Today is our lecture number 22 on chondrocyte, epigenetics, metabolism, and osteoarthritis. Our distinguished guest speaker today is Dr. Regis from Washington University. Before we start the introductions, just a reminder to the audience, please post your questions on the question answer tab on Zoom. Rob.
Thank you, Mohit. Well, it's a great pleasure to have Dr. O'Keefe with us today. Regis is the Fred C. Reynolds Professor and Chair of the Department of Orthopedic Surgery at Washington University School of Medicine. He is an orthopedic surgeon specializing in MSK oncology and reconstructive surgery of the hip and knee. Regis himself did his medical training largely at Harvard Med School and his surgical internship, and then started a journey from Rochester and Boston back and forth, went to Rochester for a while, completed his orthopedic training there, then returned to Mass General for a fellowship in surgical oncology, and then went back to Rochester where he was a member of faculty and became chair of the Department of Orthopaedics, and then moved out to St. Louis where he is currently the chief of orthopedics at Washington University. He's had an extraordinarily productive career and a distinguished leadership career. He's past president of the ORS, past president of the American Orthopaedic Association, president of the U.S. Bone and Joint Decade, and has been very active and influential at the NIH throughout his career. So, Regis, it's a privilege having you with us here today. Welcome.
Thank you, Rob. Thank you for that very kind introduction. I want to thank you and Mohit for this opportunity to join really an exceptional seminar series. And I congratulate you on just really a long list of distinguished speakers. So I'm going to speak about chondrocyte epigenetics, metabolism, and osteoarthritis, and just give a broad overview of my research program. So this is a bit of an outline, and we'll go through this, but I'm going to talk about epigenetics and really focus in the context of that on DMT3B and one of the downstream targets that we've identified for aminobutyric acid aminotransferase, ABAT. And then I'll talk a little bit about TGF-beta and how it is also regulated by DMT3B and how that also plays into a role in osteoarthritis. So this is really broadly my program. So I think we all know that OA is a very common disease. It has significant impact on the population, and a high percentage of the population is afflicted with this condition. Despite that, we have no medical treatments. The surgical treatment has grown tremendously over time, and here you see from the Swedish registry an increase in number of surgeries over time. And this continues to accelerate. And it's an imperfect solution. This is the first patient who came to my clinic when I went to WashU, and they had a BMI of about 50, and clearly they were not walking for several years, and I performed this fusion. So it's not a perfect solution. It's also a very expensive condition. This was in New England Journal of Medicine and looked at the cost of medical conditions in the United States for hospitalized patients. And among all the conditions, 284 conditions that they focused on, osteoarthritis, because of the volume of the hip and knee surgeries, was the most expensive. Now, we know that it's a biological process. It is influenced by growth factors and signaling molecules and mechanical forces. Systemic inflammation and aging have a role as well. And there's a balance between anabolic activity and catabolic activity, leads to degeneration. We focused in our laboratory recently on the genetics and epigenetic, and particularly in epigenetic regulation. So what is the role of genetics in the development of OA? And so if we look, this is a very nice meta-analysis that was published in Arthritis Rheumatology in 2014. And it looked at 9 genome-wide association studies. And interestingly, when you put all that data together, when you look at base changes that account for the proportion of osteoarthritis, the impact is relatively small. And what you can actually see is, in this particular paper, that that women with there's some protective effects, for example, of COL11A1 SNPs and in VEGF, there's an increase or decrease. But again, these are relatively small changes, suggesting that this probably doesn't account for a large number of people who develop osteoarthritis over the course of their lifetime. This is a study by John Laughlin, and he looked at epigenetics. And he did illumina infinum methylation 450B chip arrays. And he did it in about 73 knee patients and 23 OA patients, and had some hip controls. And he noted that there was marked changes in DNA methylation in the genome. And interestingly, one of the factors that they found occurred was a decrease in TGF-beta signaling. That's significant because TGF-beta in basic science studies has been implicated for the anabolic activity and the maintenance of cartilage homeostasis in patients and in animal models. So what is DNA methylation? There are these areas of the genome called CPG islands. They're rich in cytosine and guanine. It's less than 2% of the genome. These are short stretches, and they can undergo methylation at the cytosine sites. When in the promoter region, when it's unmethylated, there's high levels of gene expression. This is a suppressor of RNA expression when it's methylated, so it decreases gene expression. So this is in a balance, this DNA methylation and demethylation. The DNA methyltransferases methylate cytosine at the 5 sites, so 5-methylcytosine is created. And this is in balance with another series of enzymes called TETs that demethylate DNA. The interesting thing, and one of the ways that we were able to begin to study this effectively, is that there's a limited number of DNA methyltransferases. It turns out that there's 3 DNA methyltransferases. DMT1 is involved in maintenance of methylation during proliferation. The ones that are regulated are DMT3A and DMT3B. So that gave us a window in order to try to identify whether these genes in particular might have an impact on the status of methylation in DNA during the development of osteoarthritis, and if they play a role in that process. So, so this is the initial study we did, and this was just a screening study. Is there something that we want to pursue or look at? And one of my postdocs, Jay Shen, had gotten antibodies for DMT3A and 3B. We observed a very interesting finding. DMT3B is highly expressed in articular cartilage. It's not expressed in growth plate, and DMT3A is minimally expressed. So this suggested that, again, that the DMT3B is unique to articular cartilage that is a permanent cartilage compared to growth plate cartilage that is unique. So to understand whether this has a role in the process of osteoarthritis, we did the following study. We looked in 3 different models of osteoarthritis: aging, injury, and obesity. What you can see in these lower panels is that high levels of DMT3B expression in controls, in contrast with aging, injury of the joint, meniscal injury of the joint, and a high-fat diet, which each caused the onset of osteoarthritis in mice. You, in fact, do see a decrease in DMT3B. And then we observed also that DMT3B expression is reduced in human OA. So to understand this better and to see if there really is a role in the pathogenesis, we use conditional DMT3B loss of function models. And this is, again, just a summary of some of these experiments, but what we observed was that over time, there was a development of osteoarthritis in mice that lacked DMT3B. You can see the fibrillation of the joint surface here in these mice at 5 months, and again, now bone, presence of bone here, and loss of cartilage by 8 months. So there was progression of osteoarthritis, and this was associated with an increase in the Orsi score, as well as an increase in subchondral bone volume. Again, other find subchondral bone volume, again, being a hallmark of development of osteoarthritis. This mouse did not exist, so we created this mouse. We created an overexpression model of DMT3B. that we could conditionally overexpress. We confirmed that we could conditionally overexpress this DMT3B, and we used a meniscal injury model to see whether, in fact, overexpression of DMT3B, which is usually lost when you have injury to the joint, we can overexpress it. Would that protect the joint from developing osteoarthritis? And this is an experiment that we did where, again, you can see in control animals, there's a progressive development of osteoarthritis, fibrillization, loss of cartilage surface after the MLI model, whereas the gain of function of DMT3B is protective. Again, this shows the ORSI score and the cartilage area, which is preserved, and cartilage architecture as well is preserved in the DMT3B gain of function mice. So that's, that challenge with methylation is there's a lot of targets. So we wanted to address the question of, are there genes in particular that are regulated by DMT3B? And to do this, we used a combination of RNA-seq and DNA methyl-seq, then integrated that data, that large data set. What we found was that there was a lot of metabolic processes that were highly present within this regulatory, the sort of pathways that were regulated. This is by David Gene Ontology. And that in particular, when we looked at this DNA methyl-seq and RNA-seq, we found there were 44 genes that looked like they were maybe correlated with changes in DNA methyl-seq and RNA-seq. We identified one of these genes, which I've highlighted here, which is a 4-aminobutyric acid aminotransferase. And what we see in this particular situation is with loss of DMT3B, that there is an increase in the expression of ABAT. Now, to make sure that this actually occurs, we did some functional studies. The DMT3B promoter actually has a CpG island. It's 340 base pairs long. We were able to do a chromatin immunoprecipitation assay to identify that DMT3B actually binds to this promoter region. So DMT3B binds to this promoter region on this chip assay. So this looks like a functional CpG island that does attract DMT3B and is active. Then we asked the question about, if we lose DMT3B, do we reduce methylation at this promoter? And remember that methylation suppresses gene expression, loss of methylation leads to gene expression. So the loss of DMT3B would increase gene expression. And what you can see here is in controls, the unmethylated region was about 50%. It became highly unmethylated when we lost DMT3B. So again, this is a functional site that is very responsive to changes in the expression of DMT3B. So if we looked at the ABAT in particular, one of the things we wanted to understand is ABAT, which seems to be a downstream target, is in fact upregulated when you lose DMT3B, and also in osteoarthritis. And this is human articular chondrocytes. We show that in osteoarthritis, there's an increase in the expression of ABAT in chondrocytes from humans, and that this is responsive to the influence of cytokines, and here, for example, IL-1 beta. This is now in murine articular cartilage tissue that we harvested 4 weeks after MLI surgery, and I've previously shown that there's a loss of DMT3B in tissue, cartilage cells, and that loss of DMT3B is associated with an increase in ABAT, both by gene expression as well as by Western blot. So the question then is in fact, is ABAT, which is one of these 44 genes, is it functionally involved in the pathogenesis of osteoarthritis? And what does ABAT actually do? ABAT is an enzyme, and what it does is it metabolizes GABA to succinate. So, and you can see the pathways that are involved here in terms of energy metabolism. So it's involved in energy metabolism and the regulation of GABA levels, but subsequently as well, and it's also involved in the regulation of succinate and fumarate and activation of the TCA cycle. And what we observed is that when we lost DMT3B and we'd have an increase in ABAT, we actually would increase the levels of succinate and increase oxygen consumption. So we activate the mitochondria. This is just showing again the oxygen consumption rate with DMT3B. Loss of function is induced, as is maximal respiration. So ABAT actually controls mitochondrial function, and when it's increased, you have more fuel for the mitochondria succinate, and that then drives mitochondrial oxygen active utilization. And just to, again, confirm that ABAT was involved in this effect of DMT3B, we overexpressed ABAT, and we found exactly the same effect that you get with increase in ABAT. You get an increase in oxygen consumption rate and basal respiration, as well as maximal respiration. And then, the question is ABAT involved in cartilage catabolism? When we lose DMT3B and we subsequently have an increase in ABAT, we see that there's catabolism over an anabolic activity. Again, that's data that was done in detailed study in that, but this as an overview, this is a summary. So what we were able to do was use lenti-ABAT to overexpress ABAT in our primary articular chondrocytes. And what we observed is that, in fact, markers of catabolism, RUNX2 and MMP13, were induced when we overexpressed ABAT. So it not only appears that ABAT is downstream of DMT3B, but like DMT3B, it's functionally involved in this pathogenesis of RNA, of catabolism and osteoarthritis in this pathway. Now, one of the things that we were able to take advantage of is that ABAT is actually targeted. And there's a drug that's FDA approved called vigabatrin. Vigabatrin is used in children with seizures. And again, to prevent seizures, it increases the levels of GABA in the brain, and that decreases seizures. So that's how that works. So we're able to deliver vigabatrin, which blocks this conversion of GABA to succinate, and see whether that, in fact, would show that loss of function of ABAT would be protective against osteoarthritis in conditions where osteoarthritis would normally develop. And for that, again, we used, this is a control experiment just showing, in fact, that if you give vigabatrin, you can increase GABA levels in isolated murine chondrocytes. And here we harvested mouse brain tissue with different levels of vigabatrin and showed, again, as expected, increases GABA levels in the brain. This is an experiment showing, again, at 10 weeks with MLI, where you get osteoarthritis in controls after MLI, and with vigabatrin, we completely prevented the development of osteoarthritis. So this shows, again, that ABAT is important in a driver of the process of chondrocyte maturation, differentiation, and phenotypic changes that lead to catabolism and osteoarthritis. So where are we now in, we've just submitted a paper. So this is, again, high-level overview of this, but it's not feasible to deliver vigabatrin because it has side effects. It has effects on the eyes if it's used long-term. So one of the things that we wanted to look at as proof of concept was nanoparticle delivery of siABAT, and see, in fact, whether that could slow the progression of OA. And there's a lot of other data that's associated with this. This has been submitted to biomaterials and is currently being reviewed, so there's unpublished data. But these nanoparticles that we used were in collaboration with Sam Wickline, who's at the University of Florida, and Christine Pham, who is in rheumatology at Washington University. And Sam Wickline actually developed these nanoparticles. These are protein nanoparticles that are 25 amino acids in length, and they're derived from a snake porphyrin molecule. And they're altered so that rather than creating a porphyrin type effect that kills a cell, these are now able to penetrate into the cell. They go into the lysozyme, and then they release their cargo. And so there's been very nice studies showing that these penetrate. They're about 50 nanometers in size. They penetrate the joint very, very well. They self-assemble. And in this particular experiment, we used these nanoparticles, scrambled nanoparticles in the MLI model. You can see the onset of osteoarthritis here and complete loss of cartilage in some areas. And in contrast, that we have preservation of the joint with SI-ABAT. And again, we do see some progression of osteoarthritis in this model, but we're able to see this, this impact on the ORCE score. The other hallmarks of this are, I do want to mention too, we've looked at synovitis as well. And the synovitis is also reduced by SI-ABAT in these mice. So the synovitis is also impacted. So it's a global effect on the joint, protecting against the inflammatory process and development of OA. So obviously, one of the main features, again, of osteoarthritis is subchondral sclerosis. And we measure subchondral sclerosis using micro-CT. And I'll just run through this, this micro-CT analysis here, which is that if we look at the medial tibia in particular, that when we have the injury model and the saline-treated controls, that there's an increase in sclerosis. When we use the scrambled, we have an increase in sclerosis, but the SI-ABAT prevents this increase in subchondral bone sclerosis and is protective of that feature of osteoarthritis as well. So what I haven't been able to show you is that, in fact, that this is a summary for this first part, which is that aging, injury, obesity, environment, all those things that cause inflammation. We find actually that inflammation leads to a decrease in DMT3B in our cells. We think that's mediated by NF-kappa B signaling. We've done some nice studies and published that. So that leads to a decrease in DMT3B, and then that compromises homeostasis and leads to an increase in ABAT, increase in OXPHOS, and then eventually cartilage catabolism. One of the things we're investigating right now in terms of ABAT, and it's going to be part of our biomaterials paper, is whether or not, in fact, ABAT does this through regulation of mitochondria or some other feature. And we actually think right now that it may be by maintaining levels of GABA that in turn signal through some of the GABA receptors that lead to some of the protective effects for osteoarthritis. So the next thing I want to just briefly go over now is a little bit about TGF-beta and how we look at TGF-beta. When I started doing research back in the 80s, there was a new molecule called TGF-beta. There were about 25 papers, hadn't been studying cartilage. Some of the initial experiments I did as a postdoc were trying to understand if TGF-beta has some role in cartilage. So I've been studying this through my entire career. So I want to talk a little about TGF-beta in autophagy and maybe reprogramming the anabolic activity. Again, this is going to be a brief, high-level overview. So we, with loss of DMT3B, I mentioned that metabolic pathways were involved. We also found that it impacts TGF-beta and BMP signaling as well. In particular, we see a decrease in genes associated with TGF-beta signaling, an increase in BMP signaling pathway. And remember, I mentioned this, that John Laughlin's paper identified that in OA, there's epigenetic changes that lead to a decrease in TGF-beta signaling. Again, consistent with our findings, implicating a role for DMT3B, possibly in this process. And TGF-beta is necessary for articular cartilage homeostasis. These are just three quick models. This is loss of the type 2 receptor here. This is loss of the type 1 receptor. Both of those conditions lead to osteoarthritis. And then a key signaling molecule, SMAD3, also leads to osteoarthritis. So the maintenance of TGF-beta signaling is important for normal cartilage homeostasis. And one of the things that's interesting, this was a paper recently published by Anna Spagnoli, Science Translational Medicine. You can see that as mice, over time, you lose you lose a staining of proteoglycans, and you start to develop get set up a situation for osteoarthritis. And what she showed was that the TGF-beta receptor 2 actually is decreased with aging. And so is the phosphosmad2 signaling, the downstream signaling molecule. So it really implicates possibly that aging is associated with a decrease in TGF-beta signaling. This is a very important process in the maintenance of cartilage throughout your life. So one of the things that we focused on was autophagy, and this is a paper that we recently published in the Proceedings of the National Academy of Science. It was highlighted in a news article a week or two later in Science. And we examined that TGF-beta, and just as a brief summary, TGF-beta, it signals, in fact, through TAC1, activates FOXO1 and activates genes involved in autophagy, and is protective against osteoarthritis in that way. So I'll just show you briefly some of this data that is recently published, but you can take a look at it in more detail in the Proceedings of the National Academy of Science in the December issue. So it's known that FOXOs are reduced in aging in an OA cartilage in humans and mice. This is a study from back in 2014 showing the various FOXOs being reduced. And again, the key FOXOs are FOXO1, FOXO3, and FOXO4. And it shows, again, in these histology that FOXO1 and FOXO3 are reduced in articular cartilage. This is a very nice paper that was done by Martin Lotz, and he knocked out all 3 FOXOs, 1, 3, and 4, and he found that when he did that, after 5 months, he completely lost the articular surface. So FOXO is very important. The question we had is there an association between TGF-beta signaling and FOXOs, and can a FOXO protect against OA progression, and what are the critical downstream targets? So what we looked at here in an initial experiment was that, in fact, TGF-beta induces FOXO1 gene expression, does not induce FOXO3 or 4. You can see this with Western blot. And LC3 is a autophagy marker. And what you can also see is that TGF-beta induces autophagy. So that's the LC3-1 and LC3-2 protein expression observed here. So we did FOXO deletion, and FOXO1 deletion leads to spontaneous OA. As you can see here, here's controls at 7 months. Here is the FOXO deletion at 7 months, and there's clearly the onset of osteoarthritis. Now, the question is FOXO gain of function? Does that protect against OA progression? And we used the MLI model for this. And here you see the control mice with loss of cartilage. You see this protective effect of FOXO. It's reflected in the Orsi scores that you see here on the right. So FOXO1, loss of function leads to OA, gain of function be protective of OA in injury models. Now, this is a study that we did to really try to understand, is autophagy involved in the process, and how early is autophagy lost? And so this is using LC3 GFP mice. And I mentioned to you that LC3 is a key gene involved in initiation of the autophagy process. And you see that there's high levels of LC3 expression in normal cartilage, and it persists over time in a sham operated mouse. But with MLI, very early on, way before you see any phenotypic changes or structural changes in cartilage, you see there's a loss of autophagy. Within 1 week, there's a marked loss of autophagy. This persists over time. We looked at autophagy markers through various ways, and this is just one experiment where this is the FOXO loss of function and FOXO gain of function, and this is control. Controls here have high levels of FOXO1 expression, but when we eliminate, I'm sorry, this is LC3. We're looking at LC3. So this is LC3 expression. It's high in controls. It's markedly reduced if you lose FOXO1. And similarly, if you have an injury, you lose the expression of LC3, as I showed before, but this is reserved with FOXO1, a gain of function. So in the case that FOXO1 appears to control autophagy, we did a lot of studies that I won't get into that actually shows that that is, in fact, the case. But I want to just address this one final experiment here, which is that can FOXO gain of function compromise for the deletion of TGF-beta receptor 2? I showed before with those various models that you lose the TGF-beta receptors, you get complete loss of articular cartilage. Can FOXO1 gain of function compensate for and protect against that? So these are control, and again, this is looking at LC3, which is a autophagy marker. This is Safran fast screen staining to look at cartilage. So controls have very nice cartilage, high levels of LC3 expression. The TGF-beta receptor knockouts completely lose their cartilage, as we observed before, and there's limited expression of autophagy markers, LC3. FOXO gain of function, appears to be normal. And this is now where we knocked out the TGF-beta receptor 2 in combination with a FOXO1 gain of function, conditional gain of function. And what you can see is that this preservation of cartilage is maintained. So this loss of cartilage that you see here is preserved when we overexpress FOXO1, and you see a maintenance of autophagy. So this demonstrates that one of the key factors downstream of TGF-beta, in fact, is FOXO, and it probably acts through the regulation of autophagy. So this is, again, a brief summary of what I said, but I just want to finish with one other slide, which an area that we're working on now and very interested in, and just received new grant from the NIH on, which is look at anabolism, an anabolic effect. And the way that we're doing this, is we have found that TGF-beta induces exosamine biosynthetic. And it likely does it through activating certain genes, and particularly GLUT1, GFPT1, which is the rate-limiting enzyme in the hexosamine biosynthetic pathway, and SLIC25A1, which is mitochondrial citrate transporter. And anabolic activity and these cartilage nodules, and it's completely blocked if we have used this drug that blocks SLIC25A1. So we're trying to understand now the way that TGF-beta influences the anabolic process and how that's important in homeostasis, maybe involved in the development of osteoarthritis through these other gene targets. So collaborators, a number of people in my lab, Tri Wang in particular was essential for these studies. Jay Shen was the initial postdoc. He's now on a faculty and well-funded at Washington University. He's a postdoc with me when we initiated these studies. Audrey McClennan's been involved in these studies as well. We have Washington University collaborators, particularly Bo Zhang, who did a lot of the analysis. Gary Patty does metabolomics, and then at the University of Rochester, again with analysis of big data in RNA-seq and DNA methyl-seq. They were very helpful. So I appreciate the opportunity to speak, and I'm interested in any questions you might have. I know it's probably a little bit of a whirlwind, but I wanted to give you a brief overview of DNA methylation and how we're approaching some aspects of osteoarthritis in our laboratory. Thank you very much.
Thank you, Regis. That was a terrific overview. We have now questions populating on the question and answer tab. First question is by Dr. Jason Rockhill. Very nice to hear all the wonderful work you continue to do in your lab, Regis. It really is fantastic. I was wondering if you have looked at the relative contribution of ABAT, autophagy, and FOXOs in the synovium. A concern I always grapple with is the similar signaling in the synovium and cartilage, but the opposite responses in both tissues. For instance, autophagy activation in cartilage is protective, while in the synovium results in hyperplasia and a fibrotic phenotype. Do you have any recommendation on how we balance our therapeutic targeting to alleviate these divergent tissue responses?
Thanks. First of all, Jason, thank you for that question. That's a really key question. And, we have not looked in detail at synovium other than in the situation of these models of either overexpression of DMT3B that's protective of osteoarthritis and injury models, or inhibition of ABAT, where when we look at synovium, we're able to reduce synovial inflammation. So it looks like when we affect the cartilage, that in fact we are able to cause a healthier appearing tissue within synovium. Same thing in the subchondral bone. We think that's a secondary effect. We don't think these nanoparticles, for example, can penetrate through the cartilage into the subchondral bone. So the subchondral bone, we believe in this case, is a secondary effect. So we do think that the cartilage has a major impact, but the direct effects and whether they may sort of down-regulate the more positive effects of these targets is something we'll have to investigate. But I think you bring up a really important question. It's an area that we want to look at in more detail is the role of synovium broadly in this process.
Thank you.
A question from Fayez Savadi. Thank you for a great talk. What is the link, if any, between TGF-beta FOXO subchondral bone remodeling and the joint pain?
Yeah, that is a great question. So we have not looked at, what I actually showed you was, the experiment I showed you that we're publishing it was a experiment where we had no idea whether the overexpression of siRNA could block osteoarthritis using nanoparticles. The work that had been done by nanoparticles and published by Sam Wickline and Christine Pham and PNAS looked at a rheumatoid arthritis model and their ability to reduce rheumatoid arthritis by blocking NF-kappa B signaling with siRNAs delivered through siRNA. So there was no longer-term experiment that had been done. So we didn't do the really detailed studies along that way regarding whether or not, in fact, there's a difference in pain. I suspect my guess is there would be a difference in pain. We have to confirm that because of reduced synovial inflammation, number one, and number two, the reduced subchondral bone sclerosis. But we're in the process of doing those confirmatory studies now. Those are things we're actually in the process of doing.
That's great. This next question is by Dr. Frank Berry. Thanks for a fantastic talk. I think ABAT is also involved in neurotransmitter metabolism. Does this lead to unintended effects of using vigabatrin?
Yeah, thanks, Frank, and thanks for joining, Frank. That's a great question. As I mentioned, vigabatrin is FDA approved to prevent seizures in children, and it works through upregulating GABA and GABA signaling in the brain. So GABA is a signaling molecule. It binds to two receptors, GABA A receptor and GABA B receptor. So that is a side effect, and also some side effects on eye tissue as well, which again is very much a neurologic tissue. So we don't think it's feasible to deliver vigabatrin systemically. Interestingly, one possibility would be you could deliver vigabatrin, which we haven't looked at this. We're sort of going through a more molecular targeted approach, where you could deliver vigabatrin and nanoparticles into the joint as well. And those experiments certainly could be done. It could be confirmatory. So, but I think that's a really good point. Now, I just will say that again, because I didn't have enough time, we are now investigating. Initially, our hypothesis was that the increase in mitochondrial activation and oxidative phosphorylation was the driver of the chondrocyte differentiation process and led to a catabolic phenotype. We actually think now that it may be a balance between the GABA levels and the ability of GABA to bind to some of the GABA receptors, which are in fact expressed in articular chondrocytes. And we've done experiments with the agonists and the antagonists, and we're continuing some of those experiments. We think that and interestingly, we think that downstream then of that pathway, we think is autophagy. That's been shown in other areas as well, that autophagy is downstream of GABA signaling. So so that's an area that we're investigating currently.
Terrific. Regis, just actually a follow-up on your nanoparticles. So, what would be the uptake mechanism when you inject a nanoparticle in a joint, both in terms of cartilage, synovium?
Yeah, so they are, because they're 50 nanometers, they're able to penetrate the joint. As I mentioned, it's a protein nanoparticle that self-assembles. That's 25 amino acids in size. It's derived from a snake venom protein with changes so that it no longer is a porphyrin. The snake venom worked by opening up the cell membrane, and in that situation, leading to death of the cell. Instead, the particles can penetrate into the cell and enter the lysosomal compartment. Right. In the lysosomal compartment, there's release of the siRNA into the cytoplasm. I want to also mention one other thing that's probably significant is that you can also deliver mRNAs this way. So another approach that we're currently looking at is using these nanoparticles to deliver DMT3B mRNA. Because again, that pathway of decreasing SI-ABAT and increasing DMT3B RNA at the same time is a combinatory therapy that could be delivered simultaneously that might have a bigger impact.
Regis had a couple of questions. One was whether there's a significant sex effect in FOXO1 between males and females.
We haven't looked at that, Rob,
Okay.
but I think that's a good question. We've done both. We haven't observed a sex effect.
Okay. And then the addition of exogenous GABA, like if you're looking at, say, chondrocyte, hypertrophic chondrocyte transitions, is GABA alone enabling that that chondrocyte aberrant transformation?
So, GABA, if you block DMTs, if you block ABAT, you'll have higher levels of GABA. That would be protective. So we have done experiments, Rob, where we have delivered GABA. And GABA has an anabolic effect on the cells. We have done experiments now where we've delivered GABA with antagonists to the GABA receptors. And we can block that effect.
Right.
So we actually think that in part this may be related to GABA receptor signaling. The GABA receptors may be involved in some of the maintenance of homeostasis. But those are very, very preliminary studies. So I would caution that. But that's some of the areas that we're looking at now. And there's a few people at Washington University who are really strong, really focused on GABA signaling. And so we have some great collaborators as we're trying to move forward.
Regis, a fundamental question, and probably you're the best person to answer this based on your experience, both in terms of, doing orthopedic surgery and working with the animal models. Really, this is a question which comes up in almost every single talk. We do struggle with the preclinical models where we're doing, therapeutic testing. In your opinion, currently available post-traumatic models do not really replicate what we see in humans. How do you think we are evolving, or we are creating new relevant models which could actually help us look at age-related osteoarthritis? I know there's availability of spontaneous models, but is there something which we are not doing great and we need to improve, both from the basic science perspective, but also from the translational aspect?
Yeah, I think that's a great question, Mohit. And I think, particularly the small animal models, they're very fast. Osteoarthritis develops over a lifetime or many, many years. And even after following injury in humans, it takes years to develop osteoarthritis. So it's a markedly accelerated process. I think we do establish that the same genes and pathways are involved in both humans and in mice. I think the biggest challenge is how do you use this therapeutically or translationally? And, currently, if somebody comes into my office now, and Rob, probably see the same thing, and they have osteoarthritis and, they don't need a joint replacement yet, we'll inject steroids into the joint. And that's sort of hitting the joint with a sledgehammer. It's non-specific. It probably reduces protein synthesis and not targeted. And, the idea would be, but it does relieve pain. Is there the potential that someday we would have patients, and also it probably ultimately, again, studies really can't clearly show this, would be challenging to do these studies, but probably accelerate the degeneration of the joint. I don't think it's particularly good for the joint. So I think, is there a potential someday where someone would come in and you could deliver targeted therapy that wouldn't damage the joint, maybe protective of the joint and relieve pain through this kind of nanoparticle approach? I think that the biggest challenge is, how do you take somebody who's 30 years old and may get arthritis and, early on in their life at the age of 50, and you give them 20 years of therapy? That's the big challenge.
Right.
I was, your micro CT images were striking, Regis. And I'm wondering, I guess there's always been the debate about what stiff bone relates, how it changes the biology of the chondrocyte. I was wondering, which is, and certainly in our field, in AS, it's a big issue about how do we actually interpret subchondral bone marrow edema. And I wondered what your thoughts are. Do you see that in, I actually don't know, do you see that in the animal models as well as the sclerosis?
The bone edema, Rob, as you know, has been associated also with pain and inflammation. And, you really would need, you can't see it on a CT.
Right.
So you would need to have an MRI scan.
Right.
And, I think we'll probably get more insights as we do our pain studies. If there's an increase in pain, and the connection of, subchondral bone sclerosis is associated with edema, right, and it's also associated with pain. So I think if we also find that there's an increase, probably if we did MRI, and, you can do 9 Tesla MRIs and so on and get some information. But I think I would assume that probably because it reproduces the other features, you'd probably see subchondral edema.
Right.
Regis, in terms of autophagy, are you thinking of autophagy as a therapeutic target? Are you looking at directly activating autophagy through any specific therapeutic RNA agent?
Yeah, we would like to confirm. I mean, I think others have been looking in that, working in that space as well. And I think, if you just think from a scientific standpoint about what tissue would be important for autophagy, it would be a tissue where you don't have a lot of ability for regeneration. You need, because autophagy preserves the cell population. So in that context, clearly articular cartilage would be high on the list, and a tissue that needs to stay for a long time, needs to persist for a lifetime. So autophagy, just on theoretical basis, would be extremely important for articular cartilage. And so we're very interested in taking a look at that. But what we really was really shocking is how early the change or loss of LC3 expression occurs after an injury. So you don't see any phenotypic changes at that point at all in the cartilage, but autophagy is lost very early on. So that would suggest that that's a primary and key event that occurs in developmental life.
Right. So just to wait for a tissue to sort of accommodate or sort of compensate for any cellular changes, but also probably opposing cell death mechanism in this case. Because, there's some relationship between autophagy and cellular proliferation as well as cell death. People say, if you have increased autophagy, the cells tend to survive more. And when you have a compromised autophagy, the cells may undergo rapid cell death. Of course, we don't know if this is a direct relationship, but I believe that's what you would probably be observing in your models as well.
Yeah, I think I would agree, Mahit. We did look in these models as well. We did look at, unable to really go over too much data, but if you look at the papers that I've talked about, as well in the Boxer paper. We did look at apoptosis.
Right.
We look at apoptosis as well. So it's something that we consider important.
Regis, your first case and that guy with a high BMI reminded me that aging is associated with obesity, type 2 diabetes. They all seem to co-migrate together. Every time I open the New England Journal, I see a new application for the SGLT2 inhibitors. They seem to be great for congestive heart failure or chronic renal failure. And I wonder whether the sodium glucose transport pathway has some potential in the chondrocyte.
Yeah, we haven't looked at that. We know that GLUT1 is really important, but we haven't looked at other glucose transporters as well. The other thing that sometimes can be used is the MCT reporters, the lactate transporters as well, play a big role in also in tissues and healthy tissues, MCT 134. So we haven't looked at them as well, but this space of metabolism in its role in homeostasis, in a balance. We're very interested in the balance of how energy, so glucose, is balanced between use for energy and use for anabolic activity. And then obviously there's a combination of those. But, in normal circumstances, hexosamine biosynthetic pathway, only about maybe 2 or 3% of glucose goes down there. TGF-beta increases that. So it increases both the glucose transport as well as the percent that goes into the hexosamine biosynthetic pathway. And and if you block the hexosamine biosynthetic pathway, and we've had to create a lot of using CRISPR-Cas9. We have those various targets I told you, except for GLUT1, that there's actually 2 isoforms of GFPT 1 and 2, that is a rate-limiting enzyme. We've created conditional knockouts for those, and also SLP25A, A1. And so we're interested in trying to really determine, if they're key players and how important they are.
Yeah.
Regis, seems like we have no more questions. Rob, do you have any other question?
Nope, that's wonderful. Absolutely wonderful.
That's great. Thank you, Regis, for taking our time to speak today.
Yeah, it was fantastic.
I appreciate everyone's attention. If anyone has any additional questions or wants to follow up on anything, please feel free to contact me.
That was fantastic. Really very exciting.
And thank you to the audience for joining us today. See you all in 2 weeks. Bye, Regis.
See you, Regis. Thank you.